Good afternoon and welcome to this, our first of our 2021 webinars, delivered to you this again by Dr Tim Sandle. These monthly webinars will be covering all sorts of different aspects of Annex 1 to help you prepare for when it's introduced. Before we start, I'd like to go through some housekeeping with you. If you have any technical issues, please send a short message in the questions box on the right-hand side of your screen. The team will then respond to you direct and try to resolve your issue. Please be aware that we're working remotely, so sometimes there might be a delay and we may not be able to fix your issue. Also, during the webinar, we'd like to invite you to ask as many questions as possible, again using the question box on the right-hand side. At the end of the webinar, time permitting, we'll try to answer as many of these as possible. Any we don't answer or don't get around to answering we will answer offline and share them with you along with the slides from today's webinar. So I'd like to welcome you to today's webinar about covering Annex 1 and what you can expect. For those of you that attended any of our webinar series last year, welcome back and thank you for your continued support. If you're tuning in for the first time, my name's Annette Russell and I work as the Sterile Manufacturing Lead at RSSL. I work in the commercial team directly with the pharmaceutical microbiology team. We support our clients, both sterile and non-sterile manufacturers, with any of their analytical requirements. If you're interested in any of the services that RSSL can offer, those including those that aren't necessarily microbiological, please do get in contact with me via the details on the slide. Last year, Tim put together a 7-webinar series for us over the spring and autumn, and this year we're going a couple better than that. We're expanding our webinar series to 9 this year, 1 a month, usually the last week of the month, except for August when hopefully we can all go on a real holiday, COVID permitting. You can book any of these webinars through our website, www.rssl.com, and you can also access last year's webinar series, which are there for download on demand. Last year, Tim also wrote a white paper for us on Annex 1, which is again available via the website for you to download on demand. And if you want to keep up with any of the webinars RSSL are running throughout the year, please join our LinkedIn community. So as I mentioned, I work to provide a link between our clients and our technical experts in the pharmaceutical microbiology team. I love working with this team who are based at our new Wokingham site, as they are always keen to offer help and advice to our clients to support both non-sterile and sterile manufacturers and performing the analytical testing to the highest possible standards in a timely, efficient manner, which enables our clients to release product or risk assess their processes. RSSL can help your Annex 1 requirements by helping with environmental monitoring. This can include coming on site to perform risk assessments. We can supply plates, we can lay out plates, or we can just incubate and count any of your own environmental monitoring plates and identify any colonies, any organisms with a mouldy top that we've just acquired. RSSL can also help with your cleaning validation. This could be a full cleaning validation assessment of your entire process. We have access to training consultants that can help with that, but we can also provide disinfectant validation, where we can look at the effectiveness of your disinfectant processes. Annex 1 talks a lot about single-use plastics, and you will need to know the extractable and leachable profile of these. RSSL has a dedicated extractable and leachable team, so we can support a broad range of clients in this area. RSSL's core business is to provide support for all raw material testing across the pharma industry. This is especially true for the biopharma sector, where pharmaceutical methods may not be available. And of course, the microbiology team can help support with bioburden, endotoxin, sterility, and mycoplasma of these raw materials, as well as finished product. We mustn't forget that those vials and stoppers that are so important for sterile manufacturers need to be tested, and RSSL can help you with that. If you want to find out more about how we do that, I don't know, you might have heard of Rizwan Chaudhry. He's done an interview with some of our scientists, and if you go to the RSSL LinkedIn feed, you should be able to find this. We can help with any container integrity, also a key part of Annex 1, and understanding how your drugs can travel around and not get any contamination. And we can look at particulate matter that may develop in your system, in your vials. So we can look and help with that. Those who joined last year's webinar series will have heard about our new sterility facility, which is up and running now, I'm pleased to say. I'm sorry, I've got another issue with my slides have stopped working. So we have the capability of providing you with sterility. And finally, we also are able to provide you with training, which again is a key aspect of Annex 1. Now, you've had enough of me, I'm sure, and my slides have decided they no longer want to work. So I will pass you over to Tim. I'm sure many of you have heard of Dr. Tim Sandle, and many of you subscribe to his publications. Tim has over 25 years of experience of microbiological research and biopharmaceutical processing. Tim is a member of several editorial boards, and he has written over 600 book chapters, peer-reviewed papers, and technical articles relating to microbiology. Tim works for a pharmaceutical manufacturer in the UK and also finds time to be a visiting tutor at both the University of Manchester and UCL. So I will now hand you over to Tim.
Okay, thank you very much. Okay, so I hope you can see the slides and hear me okay. And it's a great pleasure to be back with you again. And for those of you who heard this, some of this presentation last year, it is a bit different. There's been a few more points of clarification. And for those who haven't, it's a chance to present where Annex 1 is going and where we are with current timeline and some of the things that we should expect, and also some of the things that we should be preparing for. Because one of the big debatable points will be just how much notice is there going to be in order to prepare for some of the changes that are ahead. So in this webinar, I'll briefly have a look at the scope of Annex 1, and then I've picked those areas of the text, and also I've had to be selective in the time that we've got because the new text is much bigger. You know, we're going from 15 pages to about 52 pages, and a huge expansion in terms of number of clauses. So I picked the bits that I think are most significant, particularly in terms of controlling contamination and in terms of protecting the product. So we have things like contamination control strategies and how that connects up with quality systems, tougher requirements for clean rooms, improved clarity for cleaning and disinfection, greater controls over personnel, and nudges towards requirements relating to aseptic processing, definitions of sterilisation, including source sterilisation, and also in terms of water systems. So let's begin by considering the scope of Annex 1. So Annex 1 is all about the manufacture of sterile products, and sure you're aware that these are products that are subject to special requirements, which are designed to minimise the risk of microbiological, particulate and pyrogenic contamination, and by pyrogen that's principally endotoxin. And it's an annex to the EU Good Manufacturing Practice. And the intention is to give clear guidance on how sterile products can be protected, and that's using the science-based, risk-based decision-making and the most effective decisions and technology. And that guidance also extends to the level of education and training expected of people, the principles of equipment qualification, design of the working environment, the clean room, and in relation to how that environment is verified through monitoring programs. And it applies to any manufacturing undertaken within Europe and any manufacturer who is exporting into Europe. And under the current post-Brexit arrangements, it covers all operations within the UK as well. And Annex 1 is significant. Application to terminal sterilisation and to aseptic processing, including all its variants in terms of blow fill seal technology and the like. And also it's the way it's embedded within European law, along with the whole raft of EU GMP. So that's traceable to Article 9, Part 4.b of the regulation. So although it's called guidance, there obviously is a legislative link in there, and not to follow Annex 1 would obviously lead to quite serious regulatory citations. Now, in terms of the overall thrust of Annex 1, then there are 5 main changes. So there is the global acceptance and the implementation of ICH Q9, which is quality risk management, and also its connection to ICH Q10, which is describing the pharmaceutical quality system. And the new draft actually contains many references to quality risk management, and in particular emphasizing that the industry needs to shift towards using quality risk management as a proactive tool, and rather using it instead of using it as something to react to things that have gone wrong and as part of a kind of attempt at mitigation. We should be looking for our risks up front and trying to deal with those before problems arise. There's also reference for the need for a holistic contamination control strategy, and that runs through most parts of the annex. It also acknowledges that there have been advances in sterile manufacturing technology, particularly as Annette said in the introduction, with single-use disposable plastic technologies and some of the issues of leachables and extractables that they present, and then also with barrier technology as well. And there's strong nudges within the text towards adopting those. There's also been attempts to correct ambiguity in the older version of the annex, and as well as bring the technology up to date. There are areas that were prone to misunderstanding or even conflicting definitions. So grade A was one example, with references to grade A conditions, grade A air supply, and so forth. And then also this extension that there are areas in the annex that could represent good practices for those involved in non-sterile manufacturing as well. So I want to have a look at some of these areas briefly. So first of all, we have this very important consideration of the contamination control strategy, and it's something that needs to be put together in great detail and such length. So that's the reason why in a subsequent webinar
we're going to focus on contamination control strategy in some detail. But in reading Annex 1, there is a very clear expectation that every organisation is required to have and to follow a formal and holistic contamination control strategy. So by holistic, that means that everything is joined together. So in essence, what happens in grade C may impact upon grade B, may impact on grade B, grade A rather, or there could be a type of raw material that's purchased that's substandard, but if that finds its way through the system. So it's about linking contamination events together and being able to stand back and take account of the big picture. So the document needs to reflect a site-wide strategy, and obviously, as the title suggests, it should be focused on minimizing contamination control, primarily with respect to sterile manufacturing. And there are a number of areas that need to be captured within that strategy. So a lot of it is linked around quality by design, particularly around the plant and the process, design and control of utilities, material control, which goes back to vendor approval, plus control of any outsourced activities, particularly sterilisation. And this includes items that are sterilised externally, that are then purchased by the organisation. There needs to be an oversight of those sterilisation methods. How validation is undertaken, particularly process validation, preventative maintenance strategies, cleaning, disinfection, and also a culture of continuous improvement is also embedded within the concept. So it's one of the reasons why we'll be focusing on that. And then just pulling out some examples of some of the things, some of the considerations that arguably need to be embedded within the contamination control strategy. And this is kind of giving a taster of the forthcoming webinar. But it's things like justifying the number of personnel who are permitted within critical areas, having a risk assessment and a mitigation strategy for the transfer of materials going into a grade A and B area, the process for isolator decontamination, aseptic processing controls and the risk criteria that come with those, consideration of minimising not just microbial contamination, but also, as I mentioned at the beginning, the risks around endotoxin and particles, because pharmaceutical parental products have to be free of visible particulates. Machine designs, controls around freeze drying, the rationale for the environmental monitoring program, including explanations about sample frequency and sample sizes, having a risk-based environmental monitoring program, about requirements for identification of microorganisms and how frequent that's going to be and what level that identification is going to be taken down to, and how that data is looked at and compared. The strategy for examining starting materials, and that's particularly important as the European Pharmacopoeia shifts away from stating exact specifications and puts that onus back onto the purchaser and how they're going to use that raw material. And also considering those areas that surround the classified clean room area. So the clean but not classified, or controlled but not classified areas, and how they are to be controlled. So we're minimising contamination ingress through those. So it could be everything from how people approach changing rooms to how material is moved from a warehouse into production facility, for example. The other kind of standout area is with quality risk management. So risk management and risk assessment feature considerably in the annex. And I mentioned earlier, these need to be proactive, and that the principle that quality should be designed from the outset as early as possible using the quality by design principles. And also the kind of expectation that the process of risk assessment is not simply confined to manufacturing. It needs to be extended to packaging, both primary and secondary, and also to the distribution of the finished medicinal product. So this is where GMP needs to link up with GDP, good distribution practices as well, because it's all about ensuring that the medicine, particularly if it's a sterile medicine, remains integral to the point where it's delivered, to the patient. So in terms of like approaching these various areas and just to illustrate the kind of concept of the holistic contamination control approach, I put on some very kind of crude diagrams of the kind of realization of an aseptic processing facility, and each one of these areas needs to be considered in relation to risk. And we have illustrations of both terminal sterilization and aseptic processing. And really the concept is to look at each one of those areas, to divide them up into zones of criticality, and to consider what the sources of contamination are, what the level of control needs to be, and what the level of sampling needs to be. And that's just a way of putting what I've kind of said into some kind of context. There's also focus on clean rooms, and obviously clean rooms are essential for sterile products manufacturing, and the annex will contain considerable detail on clean rooms. And this includes the design, classification and monitoring. And again, with design, we link again to quality by design and this idea of undertaking risk assessments in advance so that we can build quality at the outset. And the annex links more firmly with the global cleanroom standard, the ISO 14644 series. And this is especially so with selecting particle count locations. But the annex provides additional recommendations that kind of warns against too slavishly following the ISO grid-like approach, and the annex reminds us also that we may need to include additional locations based on direct risk to product. Now there is a change that the 5 micron count is no longer a requirement for classification, and that's in tune with the ISO 14644 standard, so it is optional. However, there is no change for continuous monitoring, particularly for sterile product manufacturing. So the 0.5 and 5 micron particle counts will remain very much in place for standard monitoring. And it's important not to get the classification and routine monitoring divides mixed up. And it's also interesting to see that the annex adds grade D at rest recommended particle levels for the first time. And it also puts controls around particle counters, and particularly particle counter tubing length, and stating that that should be no longer than 1 meter because of the risk of dropout or loss of particles, and that's something that becomes more prevalent the larger the particles become. So we can, based on those considerations, see that there are some slight variants now with what EU GMP is specifying through Annex 1 and what remains in the older FDA guidance on aseptic processing. So we still have the division on particle size cutoff values. We have the FDA document based on aseptic processing only, but Annex 1 covering both aseptic and terminal sterilization. The FDA document deals with in operation active clean rooms with equipment and people, whereas the Annex 1 looks at rest and in operation, and of course the grade D concept that there is no equivalent in the FDA document. So in keeping with the clean room concepts, then Annex 1 deals with non-viable particle counting. That's still a term I'm not overly personally keen on, because a standard particle counter through its light scattering and detection is not capable of distinguishing between viable and non-viable particles. So it is a total particle count, and we're reliant upon viable methods to give us an indication. Although there are new technologies emerging, spectrophotometric counters, that can create that differentiation and start to indicate what a particle may or may not be. But that is one area of the annex that I think could do with a little bit of revising. A change that appears in the annex is around a stated preference for separate cleanroom entry and exit airlocks. And this is something that in one of the early drafts appeared to be this will happen. In the latest draft, it's now indicating that that's something for new facilities, because the level of building and disruption work would be quite considerable. There's also the indicator that companies who are engaging in aseptic processing or other areas where there's open product should be looking to put in place barriers. It doesn't go as far as saying isolators or closed restrictive access barrier systems. RABs are mandatory, but it is strongly recommended. So there is this kind of inherent divide that will still be there between older facilities and newer facilities. And when older facilities drop into the aging facilities bracket, then there are some operational difficulties, and I think it's probably likely that regulators will be focusing on those aspects of the aging facility that are less well controlled, as much as they would be looking at how well a new facility was designed and constructed. There is also increased guidance on transfer of items into clean rooms. So there's mention of the need for multiple wrapping to be removing layers through successive clean room grades and then on transfer into the aseptic facility. There's mention of using a sporicidal disinfectant for that transfer basis. There's reference to avoiding materials that would generate fibers, and that particularly stands with the absence of visible particles in parental products and minimizing those items going into clean rooms. And as further control measures for how clean rooms of different grades interface, then there's a requirement that airlocks are fully working, that they are interlocked, and that there are time delays in place that prevent both doors from being open, and to allow items to be placed in the airlock to be disinfected and let the air circulate, contact time elapse, and to have alarm mechanisms in place to address situations when airlocks might fail. The sections on cleaning and disinfection have been considerably expanded, and it's particularly noticeable that cleaning is given greater prominence as the kind of key prerequisite before disinfection. And this is recognizing that a lot of disinfectants have poor penetrative ability. And there was an interesting study I was reading a little while ago, which was looking at a spore issue. And it was looking at one application of a sporicidal disinfectant, and then it was looking at different degrees of cleaning. And so where the level of disinfection remains constant, through increased cleaning, the ability to get rid of the spores was enhanced. So there's a lot to be said around the cleaning concept. The reference to disinfection rotation that's been a core part of Annex 1 for many years will remain, although the text will be expanded to describe the periodic use of a sporicidal product. So In the current version, it talks about 2 different disinfectants, by inference disinfectants with different modes of efficacy. Now it's indicating that a sporicide is required as part of that pattern. And in terms of disinfectant qualification, the importance is firmly on the surface testing, as opposed to the slightly more looser suspension testing. There is also some reference on running surface studies and for the need to do a risk assessment on the different surface materials within the facility, and also considering factors about perhaps the age of the surface, and understanding that a matrix approach might need to be adopted where similar surfaces can be selected to one another, and also a consideration of whether residues are a problem and what level of rinsing may or may not be necessary, which is going to depend upon the types of disinfectants adopted. Now, we've mentioned barrier technology, and for aseptic processing there is this strong indication that barrier technology is always superior to the conventional clean room because it forms that mechanism of separating person from product. So really that's the isolator or the closed RABs concept, and weak areas that are called out are with the setup of such devices and with the passing in of any materials, be it through DPT ports or through material transfer chambers or the like. There's also important emphasis that even though we might be using isolators for sterile product manufacturing, we need to ensure that product and indirect non-product contact sites are sterilized independently of the decontamination process, and that is due to the fragility around hydrogen peroxide, which is the most common means of decontaminating the space, and that's linked to distribution, the fact that it doesn't penetrate, and due to some of the variations that appear from cycle qualifications and the use of biological indicators and so on. There's also an important emphasis upon assessing isolators and particularly the individual glove ports for leakage as representing key weak points. In fitting with the previous points raised about cleaning and disinfection, there's again emphasis upon ensuring isolators are thoroughly cleaned before automated decontamination cycles are run. There's an importance put on understanding what the hold time is once an isolator has been decontaminated. So how long can that hold time last for? How many batches can be filled within that hold time? And often a media fill can be used to support those considerations. There's also a point that when biodecontamination cycles are run, again using hydrogen peroxide as an example, that the residuals from that are assessed so that they are not shown to adversely affect the product that has been filled. So that's something that might require some kind of R&D assessment, for example. And with RAB systems, then there's a new requirement that the post-setup disinfection must be with a sporicidal agent. And that then leads on to further consideration of the key aspects of aseptic processing under grade A conditions. This includes consideration of essential elements like defining and maintaining the critical process zone, seeing the aseptic assembly of filling equipment as one of the biggest, if perhaps not the biggest, risk area, because it's the one that has the greatest degree of human interaction and hence operator variability. the verification of the sterility of aseptic connectors, the risks around replenishing materials, so it might be putting more stoppers into a filling line if necessary, and controlling of manipulations and interventions, loading freeze dryers. So these are like kind of detailed, and then there's a loop back there to how these should be feeding into the contamination control strategy and how these different constituent elements have been risk assessed. So with aseptic processing considerations directly, there's also the clarification that the maintenance of grade A, the grade A concept, and supporting monitoring needs to be in place and performed from the point of setting up and for any other operation deemed to be critical. That the sample frequency and size must be such that all excursions can be captured. And this is one reason why there's been a shift more towards continuous or intermittent active air sampling. And the requirement that A, grade A monitoring needs to be continuous, and B, ideally should be continuous as well. There's also a focus with controlling the numbers of people, so the overall staff numbers that have access to the grade B area. So there's recommendations there that when there are auditors or where quality staff need to undertake checks, that there should be a way to do that through the use of inspection windows or things like CCTV camera systems. There's also mentioned that in terms of minimizing controls, that if there's microbiology staff who have been spending the morning in the identification laboratory, for example, doing some subculturing, then they wouldn't normally be expected to go into a clean room on the same day because of the additional contamination risk that might be presented to that clean room, which is something that can perhaps be more easily accommodated by large laboratories that can establish a environmental monitoring team, but might pose some restrictions for smaller laboratories, for example. Other revisions to the annex which impact on operations conducted within the clean room are kind of around controlling the elements of time, and time features quite often in the annex, and by controlling time, then an overall level of control is introduced. So this could be the time to set up a filling machine should be known. There should be a typical time and perhaps a maximum time, and that should form also part of the media fill. That the time once a filling line or an isolator has been set up before filling starts itself should also be controlled. The exposure time for vials, particularly at point of fill, should be standardized, and also the hold time of any sterile equipment as well. So after equipment's been autoclaved and held in integrous wrapping, how long can that be held for? And again, including that into a media fill is a straightforward way to try and capture those elements. And it also stands with media fills or aseptic process simulations, that here more time-based criteria is evident as well. So we've already had things like assessing the filling machine hold times and sterilized equipment hold times, but we also have a very strong indication that the media fill runtime should be equal to or slightly greater than the typical product fill run times as well, and something that becomes slightly more complicated when we're using isolators for campaign filling as well. The acceptance criteria is firmly established as 0, so no failures for media fills are allowed without detailed root cause determination, and it also will now state that 3 media fills must be conducted, the same that would be put in place for the performance qualification of a new filling line. So 3 media fills are required to recover a filling line that has seen a failure of a media fill, of course after appropriate root cause analysis. It also appears that with environmental monitoring that the EU GMP grade A limit will change from 1 CFU to simply state no growth. And I think this change is a reflection of the expectation that microorganisms are not typically recovered from grade A environments. And obviously that every recovery requires an investigation, but also it allows some leeway to consider alternative methodologies from the classic culture-based methods, such as the consideration of rapid and automated microparticle methods. So we have a bit more there, and also a strong emphasis on trending for the grade B, C and D clean rooms, and ensuring that alert levels have some relationship to that trend data. It's also clear that environmental monitoring samples should be selected by using a risk-based method, such as hazard analysis and critical control points, that airflow visualizations can inform about those risk methods, and that identifications are very important as well to see if organisms are surviving the disinfection process. There's also removal in reference to the grade A limit that I mentioned earlier, that the statement around average values has gone. So there is the full inference that every grade A event is serious and every grade A event needs investigation, and there's no mechanism to try and average into compliance. So I think most facilities weren't doing that anyway, but this kind of clarifies that element of CGMP that's been around for a while. It's also clear that every organism detected in grade A and grade B should be speciated, irrespective of where that stands in relation to an alert level. And there should be a strategy in place for considering how many organisms at C and D are subject to speciation. And that all comes down to the emphasis on the trending of microorganisms is sometimes just as important as the trending of the actual counts, and understanding what the microbial profile is also helps with developing the contamination control strategy. There is reference to the sterility test within the Annex 1 revision, and this is just a kind of emphasizing that the sterility test is limited because there is no universal growth medium, and we have the phenomena of viable but non-culturable microorganisms. But there is some advice about making the sterility test more sensitive, so giving thought to the sampling, things like targeting beginning, middle and end of the batch after any significant interventions, and with terminal sterilization for using knowledge from the thermometric work on the sterilizer to help to select locations, and with freeze drying if more than one freeze dryer is used, to drawing from every freeze dryer. With sterile filtration, then the PUPSIT requirement is fairly strongly embedded. That's the pre-use post-sterilization integrity testing. There's been a little bit of softening between the earlier draft of the Annex and what is currently in front of us. And this is for small volume products, then it is permissible to go down a risk assessment route in lieu of PUPSIT. This does require about having lots of detailed knowledge about the sterilisation process, the supply chain, the chance of filter failure, and so on. And there is no dropping of pupsit through any risk-based method for large volume products. With sterilisation technology, then there's expanded text about the importance of temperature control and testing that thoroughly during qualification, and advice on the numbers of temperature probes and for verification of temperature probes. And there is tighter guidance on freeze drying, on the control of lyophilizers, and how they need to be sterilized before each load, and the tighter requirements around those freeze dryers that are still manually loaded. And as part of the contamination control strategy, then there is a stronger emphasis upon constructing a rationale for sterilisation, about the selection and design of equipment, understanding the cycle, understanding the sterility assurance level, the approach to validation, the matrix approach that's been adopted for determining those autoclave loads that are going to be qualified, and through the using appropriate biological indicators. One of the technological shifts is towards single-use disposable technologies, and this is encouraged but with caveats that the sterility is appropriately assessed, and as Annette said at the beginning about the importance of leachables and extractables, particularly when they come into contact with the actual product. And also with the way that polymers are designed as well, particularly if they're going to be subjected to any heat or any damage that might arise from those polymers during the storage periods. With water systems, then there is a tie-in with the changes to the European Pharmacopoeia. So it acknowledges that WFI can be produced by reverse osmosis. However, there is emphasis and warnings around reverse osmosis, and that it is not as robust as distillation, because there is the weakness if biofilm buildup develops on the filter and of endotoxins as well. So it's not saying that RO is no good, but the number of controls and care and inspection of RO systems is always going to be higher as well. There is also reference about water sampling to make sure that worst case sampling points are always included, so things like the end of distribution loop returns, and that there must be water sampling taking place every day of manufacturing. So it doesn't necessarily need to be every point, and that becomes impractical for larger facilities, but there needs to be some level of sampling taking place as the water system is being used. And that not only includes microbiological testing, but needs to extend to total organic carbon and conductivity testing as well, and also controls around clean steam and steam quality. And that extends quality risk management to water systems and to all utilities in total, and that also includes reference to ensuring that there are suitable diagrams and knowledge about the system, and that any changes or add-ons are fed back to the original design concept. The controls around people have also been expanded. This includes control of personnel numbers, controls around gowning, which are much more detailed, and the prohibited no outdoor clothing is allowed to be presented to grade C clean rooms. So there's the prohibitions around that, greater emphasis upon hand washing, and the importance of visual checks for gowning. There are a few references to container closure integrity testing and ensuring that vacuums are maintained, and this forms part of both stability studies and for distribution studies using things like die-ingress tests. So for example, if you do send products to Australia, you need to demonstrate that they can go to Australia in the hold of an aircraft under cold temperatures and higher pressures and not break that integrity. Visual inspection is a concept that hasn't been covered in the annex in any great detail. It's clarified here that 100% visual inspection is expected on all products, undertaken by staff who know what they are looking for and who themselves are subject to regular tests for eyesight, for looking for particles, hairs and turbidity and so on. So that's a rapid run through some of the aspects of Annex 1. So obviously wasn't time to look in everything, but as Annette mentioned, the white paper goes into a lot more detail and give you a lot of information there as well. So just to summarize, the revisions to Annex 1 are about embedding quality risk management into the process, utilizing quality by design, about developing a contamination control strategy, about nudging the pharmaceutical sector to adopt new technologies designed to reduce product contamination, particularly around barriers and single-use disposable technologies, to make things clearer in terms of what's expected in terms of contamination control, and to really embed risk-based joined up thinking. So thank you for your time, and I can pass you back to Annette and we can take some questions.
Thanks, Tim. That was really good to hear the overview again. I'm beginning to get an understanding of how complex Annex 1 is now. I've had a couple of questions come in. If I can ask a general one first of all, people are querying when we might see Annex 1 coming in. Do you have any ideas, feelings?
I understand that obviously the coronavirus situation has slowed things down a little bit, and there was also the transfer of who was running it through Brexit, which went from UK to France. And I also understand there is some cross-industry discussions taking place about the implementation time or grace period that will be in place. So will it get issued and it's mandatory from day one? Will there be people be given 6 months or 2 years? So all that's going on, but I still understand the expectation is still at some point this year.
So I've probably got time for just 2 quick questions, I think. So I've had a question from Kathy and Gilla about what is considered as a small volume when talking about PUPSIT.
Okay, so unfortunately that's not defined in terms of the quantity, but by small volume it's the volume that will be passed through the filter, not the volume that ends up in the actual vials. And I know that obviously there's a lot of complexities around PUPSIT. I think it comes down to your own definition of what you believe small volume is, and then engaging in conversations with the regulator. So to my mind, you know, that's probably things of 25 litres or less, but that's just my view. Regulator might have a different take on that.
And the very last question we can squeeze in. So are rapid methods mentioned in the draft? Cameron wants to know.
Yes, but nothing is specified. So it's that emphasising the regulatory acceptance for rapid methods, and an indication in terms of the overall technological aspect that as rapid methods start to develop and they become more commonplace, then consideration should be given to adopting those methods. And I think the first ones that come along will be the evolution of the spectrophotometric counters, which can differentiate between non-viable and viable particles, so that they're more likely to come online. It's always a grey area. I do remember one MHRA inspector saying to me how they define the C before GMP, the current GMP, and it's sort of once we've seen something in 50% of the places we've inspected, it then edges much more into what suddenly becomes expected in more places. So it's an evolutionary concept.
Right. Well, we've certainly got more questions to answer, but we've run out of time, I'm afraid. So thank you again, Tim, for your time. Thank you for everybody for attending the webinar. I hope you found it useful, and we appreciate the questions that were asked. As I mentioned, we haven't been able to answer all of those questions, but I'll forward them on to Tim and we'll get the answers out along with these slides and a link to the recording so you can watch it again. Remember, this was the first of our 9 webinars. The next webinar is on the 28th of April. It's about the quality risk management for pharmaceutical element of Annex 1. So look forward to seeing some of you then. On behalf of RSSL Sterile Manufacturing team, I'd like to again thank Tim for continuing to deliver such informative webinars for us, for everybody out there for listening. And if you have any questions or any queries, please do feel free to contact me. Thank you.
Thank you.